Welcome to medscan Express.
A program from White Coat Hackers Media.
Yeah, where we take these complex scientific studies, the ones full of jargon, and we really try to break them down for you.
Made them easy to understand.
Exactly. Think of us as your guide through the latest medical breakthroughs. So today we're digging into some really interesting research. It comes from a collaboration involving Seoul National University Bunding Hospital.
That's right.
And it's all about predicting chemotherapy response before surgery in a specific type of bladder cancer.
Muscle-invasive bladder cancer, or MIBC.
Right. The study title is a bit of a mouthful. Predicting response to neoadjuvant chemotherapy in muscle-invasive bladder cancer via interpretable multimodal deep learning.
Yeah, that definitely sounds technical.
But the core idea, I think, is pretty exciting and potentially very impactful.
Absolutely. We're looking at a new way, really, to figure out which patients with this MIBC, that's where the tumor's grown into the bladder muscle wall, who will actually benefit most from chemotherapy given before surgery.
And that chemo before surgery, that's called neoadjuvant chemotherapy, or NAC.
Correct. NAC is a really critical question because the current standard involves this NAC followed by, well, a pretty major surgery.
Radical cystectomy, RC, removing the bladder.
Exactly. And while it can be curative,
It comes with significant downsides, right? The study mentioned some stark numbers.
it did. They talked about mortality rates, somewhere between 0.3% and 5.7%, just associated with the surgery itself.
Which isn't insignificant.
Not at all. And maybe even more striking, about 64% of patients experience postoperative complications. That's almost two-thirds.
Wow. And even with the chemo beforehand, the NAC, it doesn't guarantee success.
No, unfortunately not. The study points out that only about 35%, roughly a third, of MIBC patients actually achieve what's called a complete pathologic response, or PCR.
PCR meaning no detectable cancer left after the treatment.
Precisely. No residual tumor found when they examine the tissue after surgery. And achieving that PCR, well, it's a huge indicator for long-term survival.
How strong is the link?
Very strong. In their analysis, patients who got that PCR had a 90% 5-year survival rate.
90%. That's fantastic compared to...
Well, significantly lower for those who didn't achieve PCR. So you can see why predicting it is so important.
Right, because the current approach is more one-size-fits-all. Everyone gets the NAC.
Generally, yes. That's the standard pathway.
Mm-hmm.
But given the surgery risks, the complications, and the fact that NAC only leads to PCR in about a third of cases.
You want to know up front who that third is likely to be.
Exactly. It's all about moving towards personalized precision medicine. Can we predict who will respond well, and maybe just as importantly, who won't?
Because if you know someone likely won't respond well to the NAC.
You could potentially spare them the toxicity of that chemo, avoid delays, and maybe move straight to surgery or explore other options sooner.
Makes sense. Minimize the burden, maximize the chance of a good outcome. But predicting this is tough, isn't it? Something about tumor heterogeneity.
It's a major challenge. These tumors are incredibly diverse. Not just how they look under the microscope, the histology.
But also at the molecular level, different genes being active.
Precisely. Both molecularly and histologically, there's a lot of variation between tumors, and even within a single tumor. That complexity makes accurate prediction really, really difficult.
And people have tried before, right? The study mentioned other attempts.
Yeah, definitely. Researchers have looked at everything from germline biomarkers, different ways of classifying tumors based on stains, specific genes involved in DNA repair, imaging techniques like radiomics, gene expression patterns.
A lot of different angles.
A lot. But so far, nothing has emerged as a really robust, consistently accurate method used routinely in the clinic.
Okay, so that sets the stage for this new study, and let me try it again. Interpretable, graph-based, multimodal late fusion deep learning framework, GMLF.
GMLF, yeah. Let's stick with that.
Okay, GMLF, what makes this different? What's it actually doing?
So think of it as combining different types of clues. It's a multimodal approach. It doesn't just look at one thing.
Multimodal meaning multiple types of data.
Exactly. It integrates information from, one, the standard histopathology images, those H&E stain slides pathologists look at.
Okay, the visual appearance of the tissue.
Right. Two, it analyzes cell type data, identifying the different kinds of cells present. And three, it looks at gene expression profiles from RNA sequencing.
Which tells you which genes are switched on or off and how active they are.
You got it. So it's combining the visual structure, the cellular makeup, and the underlying genetic activity.
But where did they get all this detailed information?
It came from a clinical trial data set, the SWOG S1314 Coaxen trial. So real patient data.
Right. And the goal of this GMLF model is to take all that data
And find hidden patterns, new markers, histological, cellular, molecular, that can effectively predict who is going to achieve that complete response, the PCR, after getting NAC.
And did it work? Did they find useful markers?
Well, the results suggest yes. The GMLF model was able to identify key gene signatures that seem to be driving its predictive power.
Any specific genes that popped up?
They highlighted 3 in particular: TP63, CCL5, and DCN.
Okay, can you give us the simplified version of what those do?
Sure. Roughly, TP63 is involved in how cells, especially epithelial cells like those lining the bladder, develop and differentiate.
Okay.
CCL5 is a chemokine involved in recruiting immune cells, so it relates to the immune response within the tumor environment.
Immune system. Got it.
And DCN, Dickerin, is involved in the extracellular matrix, the sort of scaffolding that holds tissues together.
Interesting. So development, immune response, tissue structure, finding these could really help fine-tune treatment.
That's the hope, optimizing strategies based on these kinds of biological indicators.
So how does this model actually crunch all that data? You mentioned huge images, gene expression.
Yeah, it's a massive amount of information. They had 182 gigapixel whole slide images. These are incredibly detailed digital scans of the tissue.
Gigapixel.
Right. And gene expression data from 180 patients. To handle this, they use deep learning.
AI, basically.
A sophisticated type of AI, yes. It's designed to find really complex, subtle patterns in huge data sets that humans might just miss. For the images, they used a specific tool called SlideGraph plus GAY.
SlideGraph plus GAY.
Yeah, it's clever because it doesn't just look at pixels. It analyzes the spatial relationships, how different cells and structures are organized relative to each other, like mapping out the neighborhoods within the tumor.
I see. And the gene data?
That was analyzed using a different type of neural network suited for that kind of data. But the key is the late fusion part of GMLF.
Meaning it combines the information at the end.
Exactly. It processes the image data and the gene data somewhat separately first, and then integrates the insights from both streams to make the final prediction about NEC response.
And did combining data types actually work better than just using one?
Significantly better, according to this study. The multimodal GMLF model outperformed models that only use histology data or only use gene expression data.
Which really underscores the value of looking at the whole picture.
Absolutely. Cancer is complex. You often need to look at it from multiple angles to understand it properly.
Now, a prediction is one thing, but doctors often want to know why, right? The interpretable part of the name. Did they figure out how the model was making its decisions?
Yes, and that's crucial for trusting and potentially using these models in the clinic. They use a technique called SHE. It stands for Shapley Additive Explanations.
SHE.
Yeah, SHE analysis basically helps you understand which features, which specific genes or visual patterns in the images contributed most strongly to the model's prediction for a given patient. It opens up the black box a bit.
Okay, so what did the SHAP analysis reveal? What was driving the predictions?
Interestingly, the gene expression data overall seemed to carry the most weight in the model's predictions.
So the genetic activity was the most informative.
It seemed to contribute the most, yes. And within that gene data, certain biological pathways stood out. Things related to basal differentiation, how cells mature, and the presence of myofibroblasts, which are specific connective tissue cells, were significantly linked to response.
And what about the histology side? The images?
The SHAP analysis for the images also yielded insights. For instance, in regions linked to a complete response, the model noted things like an increase in the count of cancer cells and connected cells.
More of the self.
Right. But also a decrease in necrotic, or dead cell count.
Yeah.
And an increased tumor stromal ratio, meaning more tumor tissue relative to the supporting tissue.
So it's picking up on subtle changes in the tumor's microenvironment.
Exactly. The complex interactions happening within the tumor itself.
What about that tumor heterogeneity you mentioned earlier, the variation within a single tumor? Did the model account for that?
They did look into it. They tried to quantify this intratumor heterogeneity, or ITA, using different methods based on both the histology and the gene expression.
And did they find a clear link between how varied a tumor was and how well the chemo worked?
Not a direct, clear-cut link across the board. It wasn't as simple as more varied means worse response or vice versa, based on their measures. However, they did note a trend. For one of their heterogeneity measures, tumors with lower variation seem to be associated with slightly better model performance, suggesting maybe it's easier to predict response when the tumor is more uniform. But it needs more investigation.
Okay. And like all studies, this one must have limitations, right? What are the next steps?
Definitely. The big one is external validation. This model was developed and tested using data from one clinical trial cohort.
So it needs to be tested on different groups of patients.
Exactly. Data from different hospitals, maybe different populations, to make sure it works generally and isn't just specific to the initial group. That's critical before it could ever be used clinically.
Makes sense. Anything else?
Well, the model points towards the importance of certain genes or pathways, but it doesn't definitively isolate a simple set of biomarker genes you could easily test for yet.
It's more complex than just test for gene X.
Right. And they also acknowledge they didn't include all possible data types. Maybe adding other imaging like MRI or PET scans or data from liquid biopsies like circulating tumor DNA could make predictions even better in the future.
So more research needed.
Always. And experimental validation to really confirm the biological roles of the markers they identified.
Oh, yeah, let's bring it back to the listener. Why does this research matter now? What's the takeaway?
I think the big takeaway is the potential this represents for patients with muscle-invasive bladder cancer. It's a tangible step towards more personalized treatment.
The possibility of avoiding unnecessary chemo, maybe even avoiding that major bladder removal surgery for some.
That's the ultimate goal. Reducing side effects, reducing the burden of treatment, and potentially improving outcomes by getting the right treatment to the right patient from the start. It offers hope for bladder preservation strategies in select patients, too.
And it highlights the power of these advanced computational tools like deep learning, right? Analyzing incredibly complex data to find insights we couldn't see before.
Absolutely. It shows how AI can help us unravel the complexities of diseases like cancer and push towards that ideal of precision medicine. It's not magic, it needs careful validation, but the potential is definitely exciting.
It really is a significant step towards treating bladder cancer more intelligently.
Yeah, and the multimodal approach itself is key. Looking at the tumor from the outside and the structure and the inside out the genetics gives such a richer understanding.
That was a deep dive into some truly groundbreaking research on predicting chemotherapy response in muscle-invasive bladder cancer.
We've unpacked how this sophisticated GMLF deep learning model is starting to identify key indicators that could, down the line, really revolutionize treatment for this challenging disease.
Understanding studies like this, even the complex ones, brings us all closer to a future where treatments are truly tailored, maximizing good outcomes and minimizing the tough parts.
It's about making medicine smarter and more patient-focused.
Absolutely. So if you found this summary insightful, if you appreciate these efforts to make science accessible, please do like this content.
And subscribe to the medscan Express channel. We'll keep bringing you concise breakdowns of important medical research.
Thank you for joining us on this deep dive today.
We look forward to sharing more cutting-edge findings with you next time.